Welcome, dear students. Today we are going to learn about the growth and development of cotton. The cotton plant is a perennial, has an indeterminate growth habit and is reputed to have the most complex structure of any major field crop. Its indeterminate growth habit and sympodial fruiting branches result in a four-dimensional occupation of space and time that is difficult to analyze. Furthermore, these characteristics ensure that vegetative and reproductive growth is occurring simultaneously. An understanding of cotton growth and development in commercial production is important in the continuing efforts of growers to produce lint and seed more efficiently and profitably. Plant development in cotton proceeds through a number of stages, which may be divided into four main stages. Number 1. Germination and seedling establishment. Number 2. Leaf area and canopy development. Number 3. Flowering and ball development. And number 4. Maturation. Each stage involves the elaboration of new morphology and anatomy, and the transitions between these successive stages are subtle and not always clearly distinguishable. If growers are aware of these stage-dependent differences and requirements in cotton growth, then many problems in crop management may be avoided, potentially resulting in increased yields and profits. To understand each of these stages, it's needed to have a knowledge of the morphology of the cotton plant. Now we will learn about the germination and seedling establishment. Seed germination and seedling emergence are the foundation of the total crop. Inside of the cotton seed, there are two well-developed cotyledons, and located between these cotyledons is a structure referred to as epicotyl. The next distinguishable part is the hypocotyl. This is the first plant part seen above the ground in the emergence process. The tip of the small seedling, referred to as the radicle, emerge from the seed coat and ultimately form the root structure. Once the seed is placed in the ground, the seed germination begins, with moisture from the surrounding soil seeping through the seed coat at an area of specialized cells referred to as chalaza. The water softens and penetrates the tissues and triggers a wide range of chemical reactions, and the swollen embryo appears ready to burst. The radicle forces it way through the tiny opening at the micropyle end and pushes downward into the soil. At the same time, the hypocotyl also begins to stretch and forms an arch and makes its way through the soil surface. Exposed to the light, the newly unfurled seed leaves turn green and begin to manufacture food until the true leaves take over. Soon the bud above the cotyledons enlarges and unfolds to form the stem, and the true leaves and branches develop on the stem. Under favorable conditions, seedling emerge can occur in 4 to 5 days after planting. Emergence will take longer if seeds are planted in cool soils. Temperatures below 60 degrees Fahrenheit are detrimental to the germination, emergence and seedling growth. Seeds should be planted at the depth of 1 to 2.5 inches, depending on the soil type and availability of moisture to get a good stand. A good stand refers to the number of healthy, vigorous seedlings that are evenly distributed in the field. Cotton should be planted in well-prepared seed beds that are firm, warm and moist. Planting should be based on soil temperature and the weather outlook for a month after planting should be taken care of. The factors that delay germination and seedling growth also encourage seedling diseases and insect problems. The first challenge encountered by the developing cotton plant is seedling disease complex made up of one or more soil-borne fungi such as Pythium and Rhizoctonia. Treating planting seeds with fungicides often helps ward off seedling diseases. The cotton plant is constantly manufacturing new specialized cells to form the organs that carry out growth and reproduction. The 4 organs are the roots, stem, leaves and fruits. Dear students, now let us discuss the root. The cotton plant has a primary or main taproot with many branches known as lateral roots, and along the lateral roots are root hairs. After seed germination, the taproot grows downward for several days without branching. It may reach a depth of 9 inches by the time the cotyledons or seed leaves have emerged from the soil. Branching of the taproot begins about the time that the cotyledons are lifted above the soil surface and the seed leaves begin to unfurl. Soil type and texture, moisture and aeration determine how deep taproots penetrate. A few will grow as deep as 8 feet. The total root length varies by varieties and species, in addition to growing conditions. On average, roots make up 1/5 of the dry weight of the plant. Small fibrous roots start to die as the plant enters into its heavy ball loading stage and beyond. Roots grow rapidly during seedling development and nearly attain their maximum length by the time the plant enters the reproductive stage. Root depth is almost 6 times the height of the shoot at 30 days after planting. At this stage, the lateral growth is only 2.5 to 3 times the height of the plant. In most cultivated varieties, 40% of the water needs is absorbed by the top 1/4 of the root system, 30% by the second quarter, 20% of the third quarter, and 10% by the fourth quarter. Next, we are going to study the shoot system. The shoot system of the cotton plant is made up of main axis, stem, leaves, buds, branches, floral buds, flowers, and balls. The prominent main stem or primary axis results from the elongation and development of the terminal bud or shoot apical meristem. The fully developed cotton plant has a prominent, erect main stem consisting of series of nodes and internodes. The first leaf to develop above the cotyledons is referred as the first true leaf. The main stem has an erect, indeterminate monopodial growth habit. The number of nodes, the length of the internodes, and the number and location of the branches is determined by genetic and environmental factors, including cultural practices. The shoot system of a dormant cotton seed typically consists of the paired cotyledons, the primordium of the first true or foliage leaf, and the dome of the meristematic cells, the shoot apical meristem. Cotton shoot apical meristems are quite typical when compared to other dicotyledonous plants. The portion of the axis above the cotyledons, the epicotyl, is extremely reduced and it comprises a single shortened internode and one node. Leaf initiation, expansion, and internode elongation result in an unbranched plant with 4 or 5 expanded internodes about 1 month after planting. However, shoot growth during the subsequent 4 weeks is relatively rapid, so that by approximately 65 days after planting, 9 to 10 mature nodes and 5 or 6 elongated sympodial branches are present. Dear students, now let us talk about the nodes. The main stem of the cotton plant comprises many nodes, each capable of producing a branch. Except under very rare conditions, each node above the numbers 5 to 7 produces a fruiting branch. The internodal length is a genotypic character, but depends highly on growing conditions. The 3 main factors responsible for longer internodal length are the frequent irrigation or rain, excess supply of nitrogen, and inability of the plant to retain fruit. The internodal length is reduced due to a shortage of water and other stresses. The number of nodes produced decrease with a delay in the sowing season. For physiological assessment and monitoring purposes, nodes are usually numbered from bottom to the top of the plant. The cotyledonary leaves from the first node on the main stem is the only node that never has a branch on it. The first node above the cotyledonary leaves is always longer. The next nodes have almost equal internodal length, and then the length starts increasing and ultimately decreasing, close to the termination of growth. Dear students, we will now study the branches. Branches on the main stem are located in a spiral order. angled along the main stem. The cotton plant has two types of brants, monopodial and sympodial. Some genotypes may have both monopodial and sympodial brants, while others may have only sympodial brants. Monopodial brants are larger than the sympodial brants. The distance between the last monopodial branch and the first sympodial branch is only 1 internodal length. The brants that do not bear fruit directly are the monopodial branches. Monopodial brants are also called vegetative brants and are always formed at the base of the cotton plant. Monopodial brants give the plant a bushy look and usually result in a slow rate of ball formation compared to a sympodial type plant. Sympodial brants bear fruit directly, so they are called fruiting brants. The secondary brants on monopodial brants are also sympodial and bear fruit directly. Once a sympodial branch has formed at the main stem node, the plant is no longer able to produce monopodial brants above the node. The node at which the first sympodial branch will appear is a varietal character, but it is also affected by agronomic practices and treatments. Most sympodial brants are primary brants, but they may have secondary or tertiary brants. Cultivated varieties have a higher number of sympodial brants than the monopodial brants. Plants with only sympodial brants enter into the fruiting phase of growth earlier than plants or varieties also having monopodial brants. Students, our next point of interest is leaves. The cotton plant has two kinds of leaves, cotyledonary leaves and true leaves. Cotyledonary leaves have a rounded shape and have a short life. Cotyledonary leaves appear to be attached to the stem and directly opposite each other. But in fact, one is placed slightly above the other. One cotyledonary leaf falls off before the other with a margin of 3 to 10 days. Cotyledonary leaves have a maximum life of 40 days. They are thicker than true leaves and do not have pointed edges like true leaves. True leaves have pointed edges from the beginning and have 3 to 5 lobes. The cuts, known as lobe sinus, may be deep or may only be half cut. Leaves with deep sinuses provide more aeration in the crop canopy and are called okra type and super okra, depending on the size of the sinuses. True leaves reach their functional maturity in about 20 days and then begin to support other growing organs, including balls, for the next 40 days. The cotton plant has a spiral phyllotaxy, which is the arrangement of the leaves on the main stem. Each leaf is located at a 3/8 turn from the last leaf. The arrangement can be clockwise or counterclockwise in the ultimate ratio of 1:1. Gossypium barbadense has the biggest leaves among the cultivated species. A full-grown leaf is 12 to 15 cm in length and width. The leaf color in the cotton plants may be various shades of green or dark red. The upper surface of the leaf has 2 to 3 times more stomata than the lower surface, with 100 to 130 stomata per centimeter square on the upper surface and 40 to 50 stomata per centimeter square on the lower surface. Leaves may have nectary on the underside of the main rib, or they may be without nectarines. Leaves of most cultivars have hairs. Hair density varies by species and variety. However, 20 to 22-day-old leaves have the highest hair density. Leaf hairs start falling as they age. Higher hair density usually results in higher amounts of trash in harvested lint, particularly in machine-picked cotton. Leaves store potassium for use during ball maturation, when the plant is unable to meet potassium needs from the soil. The plant has highest need for potassium at the time of ball maturation. Leaves also absorb systematic insecticides and save the plant from pest damage. Spraying certain chemicals called defoliants can also enhance formation of the abscission layer. Defoliation is generally a prerequisite for machine harvesting. Our next area of focus is the fruiting brants. When the cotton plant is 4 to 5 weeks old, it will expand its basic framework of root, stem, and leaves and begin to form vegetative and reproductive or fruiting brants. The cotton plant now makes a transition from vegetative to reproductive phase and begins to develop fruiting branches. Short season varieties may set the first fruiting branch at the 4th or 5th node. Longer season varieties usually form first fruiting limbs at the 6th to 8th node. In addition to genetic differences, plant population, temperature and stress also influence the location of the first fruiting branch. Cotton plants growing close together will have fewer vegetative branches and lower fruiting branches than will cotton plants spaced out farther in the row. Excessive soil moisture and too much nitrogen early in the plant growth period also will cause the plant to set its first reproductive branch too high on the stem. The higher the first fruiting branch, the longer the plant will take to complete fruiting and to mature its balls. On the other hand, moisture, stress early in the season can result in reproductive branches beginning at nodes 4 or 5, generally resulting in a smaller plant, overall with reduced yield potential. The first part of the reproductive branch to become visible as the branch develops from the axillary bud is the floral bud, which forms the square. As the branch grows and the internode lengthens, the square is moved away from its original position next to the main stem. A leaf develops besides the square but remains very small for 4 to 7 days. Then as this leaf enlarges and unfolds, a new axillary bud is formed and develops with its own floral bud to form the second internode and square of the reproductive branch. The process continues over and over throughout the season. This type of growth results in the distinctive zigzagging form of reproductive branches. The first square is formed on the lowest reproductive branch of the plant. This branch may be located at the 5th to the 9th main stem node, depending on the variety and environment. According to research, over 70% of the total lint is produced from the first square on each reproductive branch. The second series of squares accounts for another 1/3 or more of the crop. Those squares are farther out produce less than 30% of the final number of mature balls. Dear students, our next topic is the flower. The cotton plant develops in an orderly, predictable pattern. The first white bloom appears 60 to 80 days from planting. That will be from 20 to 27 days, which is an 23-day average after the square or bud develops. It takes about 8 days between the opening of a flower on one fruiting branch and the opening of the bloom in the same position or the next higher fruiting branch. That's known as vertical flowering. About 6 days pass between the appearances of 2 consecutive blooms on the same branch, known as horizontal flowering. The cotton bloom is a perfect flower. It has both male parts with pollen-producing stamens, each with a double-lobed anther, and female parts constituting stigma, style, and ovary in the same flower. The cotton flower has 3 bracteoles inserted above the nectarines around the flower base. Bracteole size depends on genotype and ranges from 1 to 3 inches in length and width among cultivars. Bracteoles have many deep cuts known as teeth, are green in color and do not change their color significantly until the ball is ready to open. The ovary has 4 to 5 carpels or locks. Each lock contains 8 to 12 ovules that may develop into seed. The outside parts of the flower include the calyx with its 5 leaf-like divisions or sepals, 3 bracts, and 5 petals that are fused at the base. The calyx is cup-shaped with 5 teeth, indicating 5 sepals united into one. The corolla has 5 large petals tapering towards the bottom. Generally, petals are showy, white creamy in Gossypium hirsutum. The petals of Gossypium barbadens are usually bright yellow. Petals sometimes have a dark rose-colored spot at the base in Gossypium hirsutum and almost always in Gossypium barbadans. Petals are closed in a whorl until the day of anthesis. The petals of American upland cotton flowers are white or creamy colored. Those of Pima or extra long staple cotton are yellow. The flowers open during the morning and pollination usually occurs within a few hours. Pollen grains from the anther drop to the sticky surface of the stigma. The ovary comprises 3 to 5 carpels, forming the corresponding number of locules in the ball. Each carpel has a number of ovules, depending on the genotype, arranged in 2 vertical rows. The number of fertilized ovules corresponds to the number of seeds in each locule if fertilized ovules were not aborted. Stamens, which form the male part of the plant, are numerous. Pollen grains are round and have spikes. Their color is creamy or creamy yellowish in Gossypium hirsutum and yellow in Gossypium barbadans. The flower bud is the first organ that lays the foundation for lint and seed production on the plant. On average, a flower bud matures into a flower in about 25 days. Pollen grains are shed just before or soon after the petals open and pollination occurs after anthesis, but fertilization occurs 12 to 20 hours later. The threshold temperature limits for proper pollen tube growth range from 15 to 50 degrees Celsius. Dear students, now we are about to explore the development of ball and fiber. One of the microscopic miracles performed by the nature is the remarkable development of the cotton fiber. Once the tiny ovules that will become the seed have been fertilized, the young ball grows rapidly. The cotton fiber develops from the tiny cells located on the outer surface of the seed. Keeping one end firmly anchored to the seed coat, the fiber stretches out, growing longer day by day. It will reach its maximum length in 15 to 25 days, which is 18 days on an average, after fertilization of the ovule. A fertilized flower takes about 50 days to become an open ball. On average, a ball develops to full size in 25 to 30 days after flowering. Extremely low temperatures and frost dries green balls on the plant. A ball may be 3 to 5 locules and varieties can bear 5 locule balls. The size, shape and smoothness of a ball surface vary greatly among varieties. Gossypium hirsutum has the largest ball among cultivated varieties, followed by Gossypium barbadans, Gossypium arboreum and Gossypium herbaceum. Most cultivated varieties produce the following ball weight: Gossypium hirsutum 3 to 10 gram, Gossypium barbadans 3 to 5 gram, Gossypium arboreum 1 to 4 gram and Gossypium herbaceum 1 to 4 gram. The shape of a ball ranges from round to oblong and pointed. Hard lock is a condition in which a ball or part of a ball cracks but fails to open fluffy. Fibers from hard locks are weak and immature, and seeds do not have a fully developed embryo. The most costly impact is the slowdown in fiber filling, combined with poor layering of the cellulose. In approximately 24 days, the ball will reach its full maturity. An additional 24 to 40 days of favorable environmental conditions are needed for the fibers to fill with cellulose and the ball to open. Dear students, now we are going to learn about the seed. Seed size varies greatly among species and varieties. Seed size also varies within a variety. The average length and width of an upland cotton seed is about 10 mm by 6 mm. Gossypium barbadans seeds are smaller than the Gossypium hirsutum seeds that are mostly naked or have less fuzz. Gossypium arboreum and Gossypium herbarium seeds are even smaller than the Gossypium barbadans seeds, but they are fuzzy. Seed index is the weight of 100 fuzzy seeds. Gossypium hirsutum varieties have 7,000 to 8,000 seeds per kilogram. Unfertilized ovules make up the majority of moths. But some fertilized ovules may abort and form larger moths of various sizes. Seed can be stored for over 5 years if the moisture level is less than 10% and there is good aeration. The period of safe storage decreases with the increase in seed moisture content. Seed developed during the humid conditions is more prone to deterioration. Seed harvested soon after ball opening has a dormancy period called the quality conditioning period. Cotton seed is usually dormant for 3 to 4 weeks. Abscisic acid inhibits germination and the rapidity of germination. The seed has about crude oil of 16%, hull 27%, meal 46%, linters 8% and waste 3%. Lastly, we are going to discuss the fibers. Cotton fibers are epidermal trichomes or hairs that arise from the protoderm of the ovule. The protoderm is a primary meristem of the plant and consequently continues to undergo mitotic cell divisions for a number of days following anthesis. Lint fiber elongation continues for about 20 days, eventually resulting in individual cells which range in length from 25 to 35 mm, depending on the genotype and environment. Fiber length is largely controlled by the genetics and also can be influenced by the environment. Stress during this period, especially for moisture and nutrients, may cause fibers to be shorter than the normal. When the strands have stopped growing lengthwise, they begin to fill with cellulose. A cotton fiber is like a hollow tube. Each day, successive layers of cellulose are deposited on the inner surface of the fiber wall in a spiral fashion. The amount and pattern of cellulose deposited determines the fiber strength and maturity. Fiber strength is closely related to genetic makeup. Unfavorable growing conditions during the filling period may result in weak, immature fibers with low micronair, which is a measurement of maturity. The cell wall which is deposited during elongation growth is termed the primary cell wall, and it consists of pectin, callose, a thin layer of cellulose, and a cuticle on the surface. Thank you for joining today's lecture. I hope you found it informative and engaging.